Morning everyone. So let's now discuss the last topic in biochemistry and the last biomolecules inside your body, which is called protein. Basically, in our daily living, protein are those food like chicken, egg, and milk. But what are the function or the definition or the work or the process of protein inside your body? So now let's start to discuss and know what are those processes, functions, and foundation or definition of protein. But before that, let's know first what are those topics that we will be discussing. So we will be discussing the biological importance of proteins, of course, the building blocks of protein, which is the amino acid. We will be discussing also the classification of it. Aside from that, we will also discuss the introduction of peptides and of course the proteins, its classification, function, and types. Okay, so let's first discuss the introductory part of protein. It said that protein is the most abundant organic molecules of the living system. Why? Because protein has a process in the DNA replication by the name of protein synthesis. Protein is very important in DNA replication. Aside from that, in DNA transcription and translation, protein is also there. That is why it's said that it is the most abundant organic molecules of the living system. Aside from that, it also said that protein is the fundamental basis of structure and function of life. Why? Because, as I've said, protein is part of DNA replication. On how DNA replicated, protein is part of that. That is why it is the fundamental basis of structure and function of life. It also evaluated that protein is 50% of dry weight of every cell. Aside from that, it also said that it is a polymer of L amino acid or L alpha amino acid. When we say L alpha amino acid or L acid, it is a crystalline acid or stands for lysergic acid. L stands for lysergic alpha acid, or it is focusing on the crystalline acid. And when we say polymer, it is a material made of long repeating chain of molecules. As we all know, polymer is made up of long repeating chain of molecules in the structure of every biomolecule or organic compound. And it also said that 300 different amino acids occur in nature, but inside your body it is only 20 as the standard of amino acid. Okay, next to that, of course, if there is an introductory part of protein, there is also an importance or a biomedical importance of protein. It said that beside forming a long chain polypeptide in the structure of it, a protein has also a building blocks called amino acid and have its additional function. It said the very first function of those amino acid is nerve transmission. Aside from nerve transmission, it also focuses on the biosynthesis of porphyrins, purines, pyrimidines and urea. So let's focus to the three terminology. Okay, porphyrins, for the clarification of everyone, are essential for the function of hemoglobin. When we say purines and pyrimidines, we also discussed that one. It is a true nitrogenous basis. That actually purines has a function of two combining a nitrogen ring bases. Okay, what are those nitrogenous bases that is in purines? We have adenine and guanine. When we said it is pyrimidine is a one carbon nitrogen bases. When we say one carbon nitrogen bases, it is focusing on the another two nitrogenous bases, which is the cytosine and the thymine. Okay, aside from that, it also the importance also of amino acid is a short polymers of AA peptides. Okay, when we say peptides, it is a short chain of amino acid. We all know that. And if we said polypeptide, it is a long chain structure of an organic compound. Okay, so aside from that, amino acid has also a function in our system. What system, of course, a neuroendocrine system, focusing on the hormones, hormone releasing factors in the neuromodulators or in the neurotransmitters. So that are this in this system, amino acid or the protein is actually needed. So aside from that, in the microorganism, focusing also in the delta and the lysergic acid, alpha amino acid, it said that in therapeutic value, protein or the building blocks of protein, which is amino acid, is needed in the therapeutic value, in the antibiotics, and those antibiotics are called bacitracin, gramicidin, and blumicin. When we say bacitracin, it is actually used for the prevention of minor skin infection. And when we say gramicidin, it is also called as a mix of ionoformic antibiotics, or it is a type of antibiotics that is polypeptide that forms small pores of the cell membrane similar to nystatin. And lastly, blumicin. When we say blumicin, it is actually called as the anti-cancer or anti-neoplastic. It basically it basically a type of antibiotics that is used for treatment. Okay, that is used for treatment of pleural effusion or a condition of pleural infusion. Basically, it is part of, or it is actually part of the lungs of inside our body. Okay. Aside from that, it's also said that in biomedical importance of the protein, it is also needed in the some toxic or maybe toxic inside our body, or protein helps to remove some toxicity inside our body. Next, let's discuss the building blocks of protein, which is called as amino acid. Amino acid basically is a group of organic compounds containing two functional groups. The first one is the amino, which has a chemical formula nitrogen, hydrogen 2, and the carboxyl group, or basically it is a functional group of carboxylic acid that bonded in a carbonyl group and the hydroxyl group, okay, with a chemical formula of COOH. It is this building blocks of protein called a zwitterion ion process, or it has a so-called process which is zwitterion ion. When we say zwitterion ion, the structure of it is both has an acidity and basicity. Okay, why? Because the function of the acidity and basicity of it is gives the dipolar ion. When we say dipolar ion, it is an ion charge give both positive and negative charges. So this property is known as amphoteric and are often called ampholytes. When we say amphoteric, it can react both acid and base. And when we say ampholytes, it exists in the process so-called zwitterion process, in the structural formula of it. Okay. It also said that neither humans nor any other higher animals can synthesize 10 of the 20 amino acid, but inside our body we have 20 essential amino acid present inside our body. Next, we also have this classification, or the classification of the building blocks of protein, which is the amino acid. Amino acid has been classified under the various ways or structure. It actually with the side chain containing aliphatic side chain. It also, or with the side chain containing hydroxylic group, or with the side chain containing sulfur atoms, or with the side chain containing acidic group, or their amides, or with the side chain containing basic groups, or lastly, or with the side chain containing aromatic rings and amino acid. So these are those classification, or the 20 amino acid can be classified into this different side chain. And aside from the different side chain, the 20 amino acid can also be classified in polarity, if it is non-polar or polar, and of course in the nutritional basis of the 20 amino acid, if it is essential, or if it is this type of amino acid is essential, or this type of amino acid is not or non-essential. Okay, continuing. So let's discuss one by one those side chains. So let's start with the very first side chain. We have side chain containing aliphatic chains. So when we say aliphatic side chain, it is the simplest amino acid. It contains branch chain of hydrocarbon. So as you can see in this illustration, these are those type of amino acid containing a side chain of aliphatic. We have glycine, alanine, valine, leucine, and isoleucine. So in your soft copy, if you already read that one, it basically explains what are the function of this different amino acid. We have glycine, alanine, valine, leucine, and isoleucine. Okay, so in your soft copy in our discussion in your module, I've already gave you the different definition of all 20 amino acids. So to recall, when we say glycine, it is used to produce glutathione and porphyrin. It is actually a component of an hemoglobin. Next, when we say alanine, okay, alanine is an important amino acid. Why? Because it is an energy source for the liver. One of the amino acid which most easily used as an energy. So that is basically the function of alanine. Don't forget that it is a source of energy in your liver inside the body. Aside from alanine, we also have valine. Valine, leucine, and isoleucine has the same function inside your body. It's said that valine, leucine, and isoleucine has called a branch chain of amino acid. They perform the important function increasing proteins and serving as an energy source during exercise. So when you've done your exercising or workouts in your home, in your house, or in the gym, you produces the three branch of amino acid. We have valines, leucine, and isoleucine. So that is basically the function of amino acid that has a side chain containing aliphatic acid. Don't forget that in the structure of it, you can see that if that side chain is an aliphatic side chain, if in this one, this actually an illustration that basically can give you an idea that this amino acid is a type of aliphatic side chain, or it has an aliphatic side chain. So don't forget that one. Next side chain is containing hydroxylic group. When we say hydroxylic group, in the structural formula of it, you can see the formula of hydroxylic group, which is the OH. So you can see in this illustration with the serine, threonine, and tyrosine, it has an OH in each illustration or each structural formula. So don't forget. So these are the amino acid that has a side chain containing hydroxylic or OH group. Next, we have side chain containing a sulfur atom. So if in the hydroxylic group we have OH, of course in the sulfur atom we have SH or SCH3. So in this structural formula of it, as you can see, it has a side chain of sulfur atom. So don't forget, you can identify that it is or this kind of amino acid has a side chain containing sulfur if there is SH or there is a formula that is particular in sulfur atom. Okay, so as you can see literally, or as you can observe with those structure, it has the same structural formula but the side chain is different. So class, take a look Tingnan nyo. Oh, lahat ng 20 amino acid has the same structural form, but the only different of it is the side chain. Because as I've said, the 20 amino acid can be classified in the different side chain. Okay. And next to that, next to the sulfur atom, one of the classification of a 20 amino acid is actually the side chain containing acidic group or their amides. So, as you can see, if the sulfur ions give you a side chain like this, or it has a SH or SCH3 in the acidic group or in the amides, as you can see, you can eventually saw the formula or the constant formula of acidic group. So this is the constant formula of acidic group. Always remember that one, that every time you look at in the structural formula, even though those side chain has nagbaliktad-baliktad or nag-reverse sila ng pagkakalagay, it's okay. But makikita mo pa rin ang the same, ang pagkakapareha ng side chain. That is why, do not be confused if the side chain of aspartic acid is like this, and the side chain of asparagine is like this. Why? Because all of that is the same, but always remember the formula of acidic group. So that is why you have to look, you have to memorize, not actually memorize, but you have to familiarize those side chain, those constant structure of the side chain group. Okay, we have, always remember, we have aliphatic side, we have hydroxyl group, we have sulfur atoms, and we have acidic groups. These are the groups that it can actually show the classification of amino acid. Okay, so if in the sulfur we have cysteine and methionine, we in side chain or in the acidic group we have aspartic acid, asparagine, glutamic acid, and glutamine. Okay. Next to that, we have side chain containing a basic group. So always look for the structural formula containing the basic group. So this is the constant structural formula of it. And those amino acid are arginine, lysine, histidine. So as I've said in your soft copy, if you've read your soft copy, all of the amino acid has a function there. When we say arginine, it is an amino acid needed to maintain normal function of blood vessels and other organs. That is why it said that protein is the most abundant organic compound inside your body, because each member or each amino acid has a function or has a process inside your body. So that is why it is the most abundant. Basically, if you compare it to carbohydrates, it said that carbohydrates is a main source of energy lang. That is actually the function of it. But if we compare it to protein, it is the most abundant, or it is the most important biomolecules inside your body, because those 20 amino acid or those building blocks of protein, which is amino acid, can give or can help in the process of your body, in the regulations of your body. Aside from that, it is the foundation system. Why? Because protein itself can help in the DNA replication or by the process of protein synthesis. In the central dogma of our life, protein has a big impact in the process. Why? Because of that protein synthesis, it can replicate another, or it can help in the replication of the DNA. Okay. Next, we have lysine. Lysine is actually the representative of essential amino acid. It tends to be insufficient when we are on a diet centered on bread or rice. So, basically in this part, lysine is a representative of essential amino acid. When we say essential, it is a good amino acid. But it tends to be inefficient or insufficient if you eat or if you if you go on diet that is focusing on the bread or rice. Lysine is not needed there. And deficient in flour and polished rice, it actually, or lysine should be supplemented to enhance nutritional value. Why? As I've said, it is representative in essential amino acid. When we say essential, it is the nutritional value of it is good. Okay, next, or the last one for the side chain basic, we have histidine. Histidine. Okay, when we say histidine, it is an essential amino acid which is used to produce histamine and others. Okay, again, as I've said, histidine is an essential amino acid. Okay, we've already discussed those side chain. So now let's continue to another side chain containing an aromatic rings. When we say aromatic rings, the structural formula of it has focuses on the rings. Okay. And these are the amino acid containing an aromatic rings. So as you can see in the side chain of it, it's composed of rings. And as you can see, as you observe that in this type of amino acid, it also has a histidine. In the side chain containing basic group, it has histidine. Why? Because, as I've said, histidine is a semi-essential amino acid which is used to produce histamine and others. It can basically have a side chain of aromatic rings, or it can basically have a side chain of basic group. So aside from histidine, we also have phenylalanine, tyrosine, and tryptophan. Okay, these are those amino acid containing aromatic rings. As we all know, if phenylalanine is one of an essential amino acid which is used to produce a various useful amines. Okay, when we say tyrosine, it is used to produce various useful amines, and it is sometimes called aromatic amino acid together with the phenylalanine and tryptophan. And lastly, with the tryptophan, it is another essential amino acid which is used to produce various, again, useful amines. So always remember that phenylalanine, tyrosine, tryptophan, and also the histidine is one of essential amino acid. So that is actually the group or the amino acid that has containing aromatic rings. Lastly, the last classification or in the structure, we have amino acid. In the amino acid, we also have this called proline, or the amino acid of it is proline. Okay, as you can see in the side chain of it, always remember this constant formula of the side chain of it. So that focuses, or that side chain is in the amino acid. Okay. Okay, next. And aside from those side chain in the structural formula of the different 20 amino acid, of course, amino acid can be classified also in polarity and non-polar, or it can be classified in polarity divided into non-polar and polar. When we say non-polar group, these are those group that no charge on our group. Example, we have alanine, leucine, isoleucine, valine, methionine, phenylalanine, tryptophan, and purine. Okay, and in a polar group, these are the no charge on R, but no charge on R, but possess groups such as hydroxyl sulfahydryl, and amide. Okay, always remember that the differences between polar and non-polar is non-polar is not existing in the no charges of no R group, while in the polar, the same with the non-polar, but there are but. Why? Because it possesses with the other group such as called hydroxyl, sulfahydryl, and amine. Example of those 20 amino acid that it focuses on the polar group, we have glycine, serine, threonine, cytosine, cysteine, glutamine, asparagine, and the tyrosine. Okay, so that are those amino acid that is in the polar group. So let's go now on the classify, or no, let's clarify, let's clarify the word R group. So R group is the abbreviation for any group in which carbon or hydrogen atom is attached to the rest of the molecule, sometimes used more loosely to include other elements such as halogen, oxygen, or nitrogen. So that is an R group. Always remember that one, guys, that R group is the abbreviation for any group in which carbon or hydrogen atom is attached to the rest of the molecules. When we say attached, it is in the structural formula of the organic compound. Okay, so aside from that, we also have this amino acid that carries positive R group and negative R group. What are those amino acid? We have lysine, arginine, histidine, and the negative R carries a negative R. We have aspartic acid and glutamic acid. Okay, next in the classification, as I've said a while ago, that in a classification of amino acid, it also carries an essential amino acid, or there is so-called an essential amino acid, or it has an abbreviation EAA. Okay, it's said that an essential amino acid can be synthesized in the body and therefore need to be supplied through diet. Okay, it will be, or in essential amino acid, it can be proper growth and maintenance of the individual. What are those essential amino acid? We have arginine, valine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, and tryptophan. In simplest form, we can have a mnemonics in that. You can say it is AV, HEAL, MP, and TT. So this is basically the first word of those essential amino acid. For you to memorize it quickly, or for you to familiarize it quickly, you can make mnemonics. Okay, semi-essential amino acid. So semi-essential amino acid, or the adults, can synthesize of 2 amino acid and not by growing children. Oh, okay. In semi-essential amino acid, it focuses on inside the body. That basically there are 2 amino acid in this semi-essential. We have histidine and arginine. So in all 8 are essential and 2 semi-essential. So always remember those amino acid that is in semi-essential and essential. Okay? Always familiarize that. You can basically use mnemonics to familiarize it quickly or easily. Okay? Next, let's go to the properties of protein. So we've already done discussing the classification, or the classification of amino acid. We've already done discussing it. So let's now proceed to the properties of protein. So there are a lot of properties of protein. These are solubility, molecular weight, shape, and acidity and basicity. Okay, why? Because as I've said, in the building blocks of protein, which is amino acid, it has a process which is deuterium ion. When we say deuterium ion, again, it is basically a dipolar ion that carries an ion, both negative and positive charges, and it carries also both acidic and basicity, or basic of it. So always remember that one. So let's define first the solubility, or the properties, which is solubility in proteins. It forms a colloidal solution instead of true solution in water, and it has a large size of protein. So that is the solubility. Next, we have molecular weight. Molecular weight depends on the number of amino acid. Yes, the number of the structural formula, or the structural form of an amino acid, depending on it. So that is the molecular weight of it. Next, the shape. There is a wide variety in the shape: globular or insuline, oval or albumin, fibrous or elongated. Next, we have acidic and basic. Depends on the ratio of lysine plus arginine, glutamine plus asparagine. Ratios greater than 1 is basic and vice versa. So, acidity and basicity, we have already discussed that in your general chemistry. Okay, we can recall that in the acidity, it is from 0 to 7, or the neutral part. When we say basicity is from 8 to 14, 8 to 14. Right? So that is exact. When we say acid, the range of acid is from 0 to 6.9, and the 7 is the neutral, and from 7.1 to 14 is the range of basic. Always remember the pH range of basic and acidic, guys, and this is actually from the UPAC, or the International Union Applied Chemistry. So always remember that one. We can say some of the research in the Google or in the internet, it said that it can be 0.7 for the acidic, but and 7.7 for 14, 7 to 14 for basic, but eventually it corrected by the International Union Applied Chemistry, which is the UPAC, that the exact range of the acidic is from 0 to 6.9, and the 7 stands for the neutral, and 7.1 to 14 would be the basic. So always remember the differences between those research that you've read and in the IUPAC, or in the International Union Applied Chemistry. Okay? Next, we have the properties of proteins, or the charges of it. We have protein or isoelectric. Okay. It carries, of course, both positive and negative. Nature of amino acid determines to the pH of a protein. Okay. When we say pH, what do you mean by pH in general chemistry? It is potential hydrogen. Okay. Acidic amino acid, we have asparagine and glutamine. And basic amino acid, we have histidine, lysine, and arginine. And determines the charges on protein. Okay. So, at the isoelectric pH, or potential hydrogen, the protein exists as zwitterions, or zwitteric ions, and dipolar ions. Why? Because it has electrically neutral, minimum solubility, maximum precipitability, and lastly, it has least buffering capacity. So always remember that one. Next, this is actually the simplest form to classify the protein. Protein can be classified on the basis of chemical nature and solubility, function, and nutritional importance. When we say chemical nature and solubility, it focuses on the types of proteins. What are those? Simple, conjugated, and derived. When we say in the function, it focuses also in the structural, enzyme, and the catalytic, transportation, hormonal, contractile, storage, genetic, defense, and receptor. So that is the function of protein. Function of protein in a structural formula, in the enzyme and catalytic. Of course, protein is very needed in that. We have transportation in hormonal process, contractile, storage, genetic, and defense, and of course, protein can serve as a receptor. Okay. Guys, if you are very fun on watching an anime, there is a very science fictional anime. It is actually entitled Cells at Work. Just visit if you want to have a small, or if you want to have a clarification in the definition of the work of the white blood cell, the red blood cell, and the receptors. Of course, the protein is in there. You can basically watch that one because that is a science fiction. And aside from that, it can be classified in nutritional importance: complete, partially incomplete, and incomplete. Next, let's discuss that one by one. We have chemical nature and solubility, or in a simple, they are composed of only amino acid residues. They are again classified as a globular protein, or this globular protein has a division. We have globulin and albumin. When we say globular protein, it is a spherical or oval in shape, soluble in other solvent and digestible. That is globular protein. When we say globulin, it is a soluble in neutral and salt solution. Example of it, we have serum globulin. Lastly, or next to it, we have albumin. When we say albumin, it is soluble in water and dilute salt solution. So, guys, take a look with the difference between the globulin and albumin, right? Okay? Because in the globulin, it is soluble in neutral water, but it focuses in salt solution. When we say albumin, it is soluble in water, but it is soluble also in dilute salt solution and coagulated by heat. So example of that, we have serum albumin, albumin, and lactalbumin. Okay. Next, when we say continuation with the discussion of globular protein, it also have the glutalins, prolamins, and histones. Okay. When we say glutalins, it is soluble in dilute acid. Basically, the example of it is wheat and rice. In prolamins, we have soluble in alcohol. The example of it is wheat and maize. In histones, strongly basic protein, and it's soluble in water and dilute acid, but insoluble in dilute ammonium hydroxide. So basically, the example of it is thymose histone. So always take a look with that definition and function. Okay. Next to globular protein, we have fibrous protein. When we say fibrous protein, it is basically one of the simple type of protein that it is a fiber-like in shape, insoluble in water, and resistant to digestion. Yes, because if you eat fiber, you can actually not, or in digestion, it is resistant to digestion. It has three types. We have collagen, elastin, and keratin. Okay, that is the types of fibrous protein. We have connective tissues protein lacking or tryptophan in the collagen, and by heating with the boiling water or acid, it turns to soluble, which is called gelatin. Okay, next to that, we have elastin. When we say elastin, it functions in the elastic tissues such as tendons and arteries. Always remember that one. And lastly, we have keratin. Keratin is present in the exoskeleton structure. The example of keratin, of course, we've known that one. We have hair, nails, and horns. So next, and aside from those classification, those properties of protein, one of the important of the function of a protein is, of course, the four orders of proteins structure. We have the primary structure, or the secondary structure, the tertiary structure, and the quaternary structure. So the modular nature of protein synthesis are folding and folding are embodied in the concept of orders in the protein structure. So this is actually constant when it comes to the structure. It always starts with the primary structure, which it basically linked to amino acid and residues in a polypeptide chain, or in a long polypeptide chain. Second is the secondary structure, which is stable. It has a stable arrangement, and the structure of the secondary structure, or its illustration in secondary structure, is actually basically the same with the DNA. Why? Because it has a spiral structure, okay, or it has a helix structure. It is basically stable and arranged in a residue, giving the rise to recurring structural patterns. Okay, naka-bold yan siya. That is very important. Into
geometrically ordered units, which is twisting, resulting in A or alpha helix or pleated. Next, of course, the third one is the tertiary structure. When we say tertiary structure, it is not the same with the two structure. Why? Because in tertiary, it has a three-dimensional structure. Assembly of secondary structure units to form a large functional units. So it actually basically combines in a secondary units, but the structure of it or the illustration of the structure of it is not the same. Lastly, we have quaternary structure. The arrangement of the space of it has a two or more polypeptide subunits. Basically, the structural formula or the structural or the illustration of structural of the quaternary structure is complicated. Why? It like when you crumpled a paper. That is basically the illustration of quaternary. I will be showing you that one. Okay, so this is the illustration of the four orders of protein structure. We have the primary structure, the alignment of it aligned. Yan, the primary structure. We have the secondary, as I've said, it has alpha helix or it has a spiral structure, as you can see. And of course, we have the tertiary structure combining the secondary structure, as you can see in the illustration. So that is the tertiary structure. And lastly, the quaternary structure. As I've said, quaternary structure is the same if you crumple the paper. But once you crumple the paper, the illustration or the structure of it is the same with the quaternary structure. Okay, so assemble by subunits. Then, and that would be all. That would be the last topic that we have. Protein, it said to my last slide, protein your body will thank you for. Okay, why? Because protein is very important inside your body. Always remember that protein is the most abundant organic compound or the most abundant biomolecule inside your body. So thank you very much for listening and for always keep me updating with this course, and I hope you've learned something in biochemistry. And in your exam, I will not be focusing on the structure, but you have to familiarize the side chain of the 20 amino acid. And also, be familiarized with the essential amino acid. What are those amino acid that is part in essential amino acid? What are those amino acid that is part in semi-essential acid? So, meaning, literally, you have to study the classification on or how can amino acid be classified in different process. Okay? So, thank you very much, everyone. Bye.